ladies and gentlemen. Before I start, I would like to bring greetings from Colombia to the 5th annual virtual symposium hosted by the International Branch of the Entomological Society of America. Today, I'm going to make the presentation Diversity and Morphology of Green Lacewing Insecta Neuroptera Chrysopidae associated with a banana agroecosystem in Costa Rica. This research was part of the senior project of the first two authors, Garcia Quiseno, who's presenting today, and Valcárcel Minian. We also collaborated with professors Walter and Coleman from Earth University and Morera Montoya from University of Costa Rica. So let's start talking about bananas. Bananas are cultivated in all tropical areas where it present a very good yield. This crop represents a very important economical pillar for developing countries. In 2019, it was an estimated production of 21 tons to export globally, mainly from Ecuador, Philippines, Guatemala, Colombia, and Costa Rica. Based on its importance, it is vital to perform agricultural practices, including the IPM. The main quarantine pests for banana in Costa Rica are Pseudococcus elise, which is mealybug, and Diaspis boswelli, which is known as the white scale. This last one could cause very high economical losses and also high managing cost, reaching $1,000 per hectare per year. So it is important to make a focus on the integrated pest management that is developed, and it's mainly related to the use of industrial detergent, mineral oil, bagging practices, and also, and very important, the monitoring of insect pests and their natural enemies. Some of the natural enemies that have been reported for the Diaspis boswelli control has been the ones from the family of Chrysopidae, some genders, Cerocrysa and Leucocrysa. But what are they? What are those? These are generalistic predators of soft body insects such as aphid, mealybugs, and white scales. Some authors agree that we need to develop some knowledge in order to, before considering that part of IPM program. Knowledge such as biodiversity, right identification, habitat information, biology, and others. That's why we got the objective for this research as the identification of Chrysopidae species in a banana agroecosystem. And for this, we need to carry out some samplings and the identification of Chrysopidae species that were collected. And also with all of this information, we could be able to elaborate a dichotomous key for identification of them. Let's talk a little bit about the place that we were working in. It was the project number 1 at Earth University, located in the province of Limón in Costa Rica. And we were working with Williams, which is a cultivar of banana, one of the main banana types that is working in this area. And in the agroecosystem, we were provided with 4 sampling points mainly, and the samplings were carried out during the night and during the day. During the night, we were using a white cloud vertically and LED light. And for the sampling during the day, we count with 10 samplers approximately that were performing direct searching. We were looking for eggs, larvae, pupae, and adult, all of these 4 stages. And once we collect them, we place them in petri dishes individually, and we read them. The food that we provide to the larvae were Cytoturga cerealela eggs, which are lepidopteran. after the breeding process, it was very important to get mature adults because the genitalia, it was a fundamental key for the identification. So we need to get this genitalia. How we did it? How we get it? First, you cut the abdomen. You add the abdomen to a 10% KOH solution in a test tube, and you put it in a water bath. And then we remove it from it. We rinse it, and then we use a color in order to see in a better way like the structures. And then with the help of some needles, we remove all the material that were not the genitalia. At the very end, we get a genitalia structure. This one, the one that you're able to see in the image I, is a Leucocrysa amazonica genitalia of a female, and we were helped by the Dr. Sousa Duque from UFRA University located in Brazil for the identification of all the species. In total, we found a 72% in the larvae stage. We found these individuals mainly in the lower leaf superface and the pseudostem. Once we identified the species, we found a 22% of Cerocrysa cinta, Leucocrysa lancala with a 25%, and the third one more abundant was Cerocrysa smithi with 22%. According to Rugno and other authors, they also found Cerocrysa cinta as the most abundant species in other agroecosystem, and they mentioned that it was because of their resistance to some agrochemicals. There are two agrochemicals that are very used in banana with the bagging methods, and they are pyroproxyfen and bifenthrin. And these two are some molecules that Brugno and other authors mentioned. So it is very important and taking this into consideration about the resistance of against these molecules. In the following images, you're going to be able to see some characteristics of the adults that we found. We found 8 different species in total. You're going to be able to see in a very quickly way the male and the female genitalia, and then the location. So Cerocrysa smithi have been reported from southern United States and to South America. The second one is Cerocrysa cinta. This is population Mexico to Florida, and it has been reported from Florida to South America. The third one is Cerocrysa everest, reported from Mexico to South America. Leucocrysa amazonica, this is one of the largest species that we got into in the investigation, on the research. And as you could see, the genitalia of the female genitalia is also very large, especially in the spermatocecal conduct. And it has been reported from Guatemala to Brazil. Leucocrysa lancala reported from Costa Rica to Brazil, and this one is very important. Leucocrysa lateralis, because it has been reported just in 2 countries, Guatemala and Brazil. And in our investigation, we reported by the first time Leucocrysa lateralis in Costa Rica. Finally, we got 2 species from the gender Leucocrysa, and we identify them like SP1 and SP2. To conclude, we got 304 samplings. Most of them were in a larvae stage. 178 from the total were identified, and with all the information that we got, we could be able to elaborate morphological characteristics description, and also the dichotomous keys that are a very important tool for producing not only for Costa Rica, but also for Latin America and other places. So let me end by thanking you for the opportunity to share all of this biodiversity information, and I will be very glad to help with any follow-up information related to the investigation. And once again, thank you very much.